with an introduction to AIMT, that is a DST FIST sponsored institute, a prestigious award for the AIMT and for MIT University. AIMT was established in the year 2004 with 5 students enrolled in PhD and 6 in masters, and as of now we are offering 4 programs: undergraduate, postgraduate, PhD and DSc program. And these are the number of students who are presently enrolled. As you can see, graduate, postgraduate and PhD, 45 PhD plus 2 ODC Viva were conducted, and a lot of students have passed out from this institute, as you can see. And we have all the major facilities. We are equipped with all the major facilities in our institute. As you can see the list of the facilities. And this is a glimpse of some of our facilities. As you can see, modern microscopic facility, confocal microscope, fermentation, atomic absorption spectrometer, nanoparticle size, zeta potential analyzer, gas chromatograph, temperature, moisture, light control greenhouse. This is here Professor Dr. Ajit Verma being awarded for a Lifetime Achievement Award in BioAg Asia 2022 by Agriculture Today Group, awarded by Dr. Tilochan Mahapatra, DG ICAR. So, and these are the students' achievement in the last 5 years. Students have won numerous awards and fellowship, as you can see from the list. And the main motto of AIMT, we nurture talent. This is the example that how we have guided the students right from our first batch of BSc who filed the patent, got Prime Minister DST Inspire program and NASI fellowship. And student, she is presently in Germany. Dr. Shubhangi Sharma now, alumnus of AIMT, she has completed a PhD, and when Prime Minister visited Germany in Berlin, she was in the part of delegation that met the Prime Minister. And Dr. Navpreet Kaur, she is doing her job in Royal Bournemouth Hospital in UK. Khushbu Malhotra, now she is working in UK after completing her master's studies in Germany after completing her master's. Srinivas Patil won award in India International Science Festival, filed patent and made publications during his undergraduate stint at MIT. Sanskriti and Arpit, they published book chapters. Similarly, Aditya Kumar filed patent. Upasna and Josna published book chapters. Supriya filed 1 patent. And I am very happy to inform you that she is now selected for the post of Scientist C in Government of India. And Sneh Bajpayee, she filed 1 patent and now doing PhD. And Suryansh and Mehak, they also filed patent and published a paper. Vikrant Hari Kashyap, very brilliant student of 2021 batch. He was offered PhD positions in 10 countries and finally settled for University of Sheffield, UK. Similarly, very brilliant student Shubhangi Mahajan won numerous awards during her stint, and she was selected for PhD in Max Planck, Germany. Aditi Jain and Chitrakshi, they co-edited 1 book, made book chapters. Ishan Tiwari and Ritika Chauhan, still with us, filed patent during their masters. Chesta and Manisha filed 2 patents, and their technology developed by them was selected by DWIH, German House for Science and Innovation, New Delhi, as one of the top 15 innovations by young scientists. Rashmi Das filed 1 patent. Madhunita and Purva, they got Diad Fellowship. Deepika Singh got 2nd position in volleyball's championship. Ishan Tiwari and Astha Rupali, they qualified CSIR UGC NET examination. And these are the faculties who were selected for postdoc fellowships overseas. And as I speak, Dr. Kanchan Vishwakarma, she is still doing this fellowship in Sweden. This is a team AIMT led by Professor Dr. Ajit Verma, mix of experience and young scientists. And this is a support staff of AIMT. So the research focus of MIT Institute of Microbial Technology focus on enzyme technology, microbial ecology, plant microbe interaction, plant genetic transformation, plant genome editing, nanoscience, environmental microbiology, and bioremediation. So you can see the students, especially the list of faculty expertise in various areas, right from plant microbe interaction, enzyme technology, plant genome editing, so on. You can see, and faculty have won numerous awards and fellowships. And world ranking of top scientists, Professor Dr. Ajit Verma in world ranking of top 2% scientists, and Dr. D.K. Chaudhary in list of Indian scientists who were in top 2% in 2019 Stanford study. AIMT is very rich in publication and patent. We have published 368 SCI journals in last 5 years, 215 book chapters, 70 books were edited. Total books published from AIMT is 128. Patent filed 54 and patent granted are 12. There are two ongoing research projects going on. And what we do, we work on self-reliant India, that is Atmanirbhar Bharat. We have developed novel innovations to make India self-reliant. We have developed novel innovative technologies for sustainable and climate resilient agriculture. And this is the culmination of our work, the root tonic, the magic fungus, that is Piriformospora indica, whose technology has been transferred to 4 companies, including one in Taiwan. And this is a world synthetic media, as you can see. And when the fungus is applied to seeds, seedlings, tannin crops, it provides, resistance against biotic and abiotic strength, and also promotes plant growth productivity. As you can see here, difference between control and treated crops. And this is up close picture, so suppressing the disease. And another picture, you can see this fungus was discovered from Thar desert of western Rajasthan, and this is working equally well in cold desert of Leh and Ladakh at minus sub-zero temperatures. Another root endophyte we have recently discovered is from belonging to Teloromyces species HNB9, an azenically cultivated fungal strain. So this fungal strain also has shown plant growth promoting activities like zinc, phosphate, and iron solubilization, indole acetic acid production, and its interaction in greenhouse has shown positive interaction with the plants. And as you can see here, root colonizing, and its field interaction is also positive in terms of plant growth productivity. And very recently, we have developed a natural fabric out of its culture filtrate treatment, and this natural fabric is also antimicrobial in nature as per the Japanese standards, and we have filed a fresh patent for this. And another research that P. indica prolongs the shelf life of cut flowers. The cut flowers are in great demand, as you can see, these beautiful orchid flowers. And here you see 0 day, and after 35 days, you can see the P. indica treated cut flower is still fresh, whereas the control is completely senescent. So we have developed successful stories from our technology. We have transferred the technology of Rootonic to 4 companies and of HNB9 to 1 company. And we have MOU and material transfer agreement with many companies, including ICL Innovation Israel, HNB9, and Premier Tech Canada, Wacom Biotech, with whom we are having active collaboration. And this is a new product in the market, Crea DMF, that is a desert mycorrhiza fungus. So Dr. Reddy, who has developed this product, he calls it desert magic fungus because this mycorrhiza performs magic. And these are the books which were published in 2020, 2021, and 2022. And this is the activity calendar of 2023. We are going to have sub-sponsored symposia on microbe in climate change and food security that was held during 9-10 February 2023. And very eminent expert delivered their lecture on day 1 and day 2. And also we are going to, we are having DBT sponsored again symposia on climate change and food security during 1st to 3rd March 2023. Again, eminent speakers were present there. Just a minute. So thank you very much for your kind consideration. Thank you.
Thank you, sir. Now, I would like to invite Professor Dr. Ajit Verma, a distinguished scientist, Professor of Eminence, and Head of AIMT Department, to deliver a welcome address.
Thank you. I welcome you all for this faculty development program. We are going to have two lectures. One, Professor Mishra from JNU, other from Germany, who will speak just after Dr. Mishra. So you all are welcome, and you attend the lecture very carefully, and you learn a lot from these two lectures. Thank you very much, and welcome.
Thank you, sir. Now, I would like to invite Professor Dr. Ulrich Matthias for his theme lecture on molecular analysis of diatom bacteria interactions leading to carbon sedimentation in the ocean. Dr. Ulrich Matthias is currently the project head of peptide research on cocoa at Kabuse NV Jacobs University. He has done his PhD in microbiology from Max Planck Institute of Medical Research, Heidelberg, Germany. Dr. Ulrich has been honored with many prestigious awards and fellowships like the Professor of the Year Award at Jacobs University, Bremen, the Chemical Industry Foundation Fellowship, the German Academic Exchange Service Postdoctoral Fellowship, and that short-term lectureship. He is a member of the American Society for Microbiology USA, International Society for Molecular Plant Microbe Interactions USA, and the Scientific Advisory Board Botanica Bremen. A very warm welcome to you, sir.
Thank you very much. I think I'm going to start sharing my screen, if you don't mind, so that we can save a little bit of time. Let me see. First of all, dear Ajit, thank you very much for inviting me. That is a great pleasure to be here again. And I would like to express my gratitude that I have the chance to talk to young scientists at your institution, which have made considerable progress. And I'm really proud to be an honorary member of your institution. So what I would like to do today, and I hope you can see my screen, I would like to talk about one of our projects which guides us out into the ocean. This is very unusual for your institution, I guess, but it's about microbe-microbe interactions, and that's why it fits again into the scope, into the overall scope, because what I would like to show you is how you develop and then also exploit a model system without going back into the ocean all the time, but doing everything on site in a laboratory where we can investigate a couple of very fundamental processes which take place there. So as a microbiologist, of course, you can work on very different topics. I think before, a couple of years ago, I gave a talk on rhododendron, our rhododendron research, which was, of course, also very well received by the audience, because rhododendron as a plant with medical importance turned out to be quite interesting. Today, I would like to, and that was also Ajit Verma's suggestion, talk about a completely different project, which we have also worked on for a couple of years, and this would be the molecular analysis of diatom bacteria interactions. These interactions are important for the overall carbon dioxide sequestration in the ocean. What you can see here is a general, okay, let me get the pointer, laser pointer. I think that's better. What you see here is the general opinion and also the model for the so-called biological pump, the carbon pump. So fueled by sunlight, we will have photosynthesis by large and small phytoplankton. There will be nets of interaction, food webs and food nets, of very complex nature. But there will also be a lot of interactions between those small phytoplankton and medium-sized phytoplankton and bacteria, because many of the bacteria are heterotrophic, and they feed on the photosynthetic products of phytoplankton. Now, this is a common belief. when they interact with the phytoplankton, as shown here in this part, the phytoplankton also tends to clump together and form, based on exopolymers, which are formed as a consequence of the interaction of the bacteria with the phytoplankton, tend to form then so-called marine snow. Marine snow is that stuff which you can see when you watch underwater movies, which settles down very slowly, falls down like snow in the air, but this time, of course, in the bottom, to the bottom of the seafloor. This organic carbon, which ultimately enters the seafloor, is sedimented and for a certain period of time is taken out of this cycle, of this biological pump. It's about, as you can see, a very minor proportion. Most of it is getting back into carbon dioxide. There will, of course, also be a non-biological exchange of carbon dioxide between the water and the atmosphere and so on. If we have rising carbon dioxide levels in the atmosphere, we will, of course, also have rising dissolved carbon dioxide or carbon in the ocean, which leads ultimately also to an acidification, which is a very important and very dangerous process. You might know that marine water or seawater has a slightly alkaline pH. And if this alkaline pH drops into the more neutral range, a number of organisms in the ocean will not be able to survive. So that's why it's important for us to understand what drives this interaction between bacteria and phytoplankton. Now, to my surprise, when I started thinking about this project, there was no system investigating that. So there was no in vitro system, for instance, to study bacteria diatom interactions. Diatoms are these medium-sized phytoplankton. And which tells us about which genes are important, which chemical substances are exchanged, are there signaling substances, and so on. What I could find within the literature were, of course, models. Yeah, so this, what you can see here from a beautiful drawing from electronic drawing from Roman Stocker, was demonstrating how such a aggregate, which is filled with slime, debris, and all the other materials, but of course also diatoms, which were sticking together, would leave a plume of chemical substances in the back. So that prompted us, for instance, to investigate whether there's chemotactic, there are chemotaxis processes playing a role. And Roman Stocker, at that time from the Massachusetts Institute of Technology, became a valuable collaboration partner. He is now at the ETH Zurich in Switzerland. So, in other words, there was a need in the 2010s, 2012s. There was a need for establishing a model system which is genetically accessible. And we got that with the help of a number of experiments. We found out that there is a heterotrophic bacterium, a gamma proteobacterium, which we named Marinobacter adherens because we had to identify its species, and it was a novel species. We called it adherens because it was adhering to diatom cells. It's widespread in marine and terrestrial habitats, and it was originally isolated from aggregates in the German Wadden Sea, so right next to Bremen here in northern Germany. As a model partner, interaction partner, we selected Thalassosira weissflogi, which is a globally distributed diatom cell. Here you can see that it has this beautiful cylindric shape with lots of spines on both ends. And these spines, of course, are used by the diatom to float, to have a floating device in the ocean. And as you can also see in the back, these are bacteria. The bacteria also have a tendency to attach to these spines and also to the surface of the diatom. Thalassosira is also known, and that was not our achievement, but that had been known previously, that it produces transparent exopolymeric particles, so-called TEP particles, in the presence of bacteria. So the interaction model then was consequently that Marinobacter adherens is a typically attaching bacterium we sequenced the genome and we made the bacterium genetically accessible. You might say, okay, that's nothing rocket science or so. At that time, we are talking about 2010, 2011, genomes of bacteria were still relatively elaborative to obtain, and the genetic accessibility was completely unknown. You might have noticed that if you want to work with mutants, if you want to insert reporter genes and other things, then you would have to very carefully check whether this organism you are working with is actually transformable, that you can transform it with foreign DNA, or that it is expressing, for instance, reporter genes under the control of known promoters, which usually come from E. coli. So while E. coli and a few other organisms, not sure about the fungal organisms you were referring to earlier, are very easy to work with genetically, for marine organisms, with the exception of Vibrio cholerae, we have very few systems, if at all, and they are usually involved in other processes. So the what we need for the interaction is attaching bacteria and photosynthetically active diatoms. So that's what we found out by inactivating the photosynthesis and then noticing that the tap formation was not happening. We also found out by mutant analysis that chemotaxis and flagellar motility were important for the attachment. Now, also, this might seem very logical. Of course, something needs to move in order to interact, and something needs to be attracted while interacting. We had to prove it, and we could prove it by site-directed mutagenesis because we knew where the genes are, and we, of course, could actually modify, manipulate the organism. What was of particular importance is that which proteins are involved in the interaction, and there was a big surprise coming up, because if you have a photosynthetic system, a plant, or a diatom, and you have a heterotrophic a bacterium or fungus, what do you expect what the fungus or the bacterium gets from the plant? Carbohydrates, right? Carbohydrates, sugars, all kinds of photosynthesis products. What we do not expect is that the plant gives away amino acids, because amino acids are relatively secure. We need the amino acids inside the plant or in the diatom. Now, what we obtained, or what we found out, was in this analysis, and I will show you, give you a couple of examples for that, is that it's actually amino acid metabolism, which is upregulated when the diatoms are present in the bacterium. So the diatoms trade amino acids and get in return, and that is something we just found out very recently and published in a Nature Microbiology paper this February, or 2 months ago, is that in return sugars are released by the bacterium and are taken up by the diatoms in times when the diatoms are not massively acting photosynthetically. Very interesting stuff, and we of course had to investigate it. The genome of Marinobacter adherens can be split into 3 parts. The main genome, which is 42 megabases. Sorry, here it's the wrong number. It's 42 megabases. We have 2 large plasmids, one of 42 kilobases and one of 187 kilobases. So this is a mega plasmid, if you want, but it contains a number of interesting genes, including some involved in amino acid metabolism. To demonstrate you that the mutant generation can be very efficient, we mutated 2 things, namely one, the flagellin gene, which is responsible for forming the flagellum. As you can see, Marinobacter has 1 polar flagellum, which is also very common in marine bacteria. And the other gene is involved in the hook, in the flagellar hook. So now most of you, having had microbiology in lectures or in your everyday work, understand how these flagellar motors work, and that the hook is of course indispensable for the activity, and that it will twist and rotate the flagellum in a very organized manner. Now we knocked out the corresponding genes, and we obtained FLE-C. FLE-C stands for flagellin mutant, and hopefully you can recognize this. These are electron microscopes, micrographs, that the hook is still present. Whereas when we knocked out the hook, the FLE-G gene, the hook was completely absent. So with the help of these mutants, along with also some chemotaxis mutants, so we also knocked out chemotaxis, and when you compare the wild type and the GA, so-called GA mutant, which is effective in chemotaxis, in soft agar with a low nutrient concentration, then you can see a spreading of the wild type, because the cells need to find the little amount of nutrient they have, whereas in the GA mutant, we don't see this spreading anymore. So they are chemotactically inactive or unable to detect particular compounds. So with this in hand, we could now, of course, undertake a thorough analysis of the interaction. The question was, do those genes play a role for the interaction? And if so, what role? One very simple attachment assay we could do in Eppendorf microtubes here, 1.5 milliliter microfuse. This is the medium itself. And then we let the bacteria incubate, remove the bacteria, and stain with crystal violet in this particular case. Yeah? The wild type attaches to the surface, to the inner surface of the Eppendorf cup very efficiently, whereas the flagella mutants did not. Chemotaxis mutant also attached, so that was, of course, again, a demonstration that we had knocked out specific genes in a very accurate manner. So this is again the background or the phenotype of these individual mutants, and you can see that both the removal of the flagellum as well as the removal of the hook would lead, which of course also does not have a flagellum, would lead to a similar phenotype. So these mutants were then labeled, because genetic accessibility means that you can, for instance, introduce fluorescent marker genes, for instance, green fluorescent protein or red fluorescent protein or yellow fluorescent protein. And you can then visualize those microorganisms in confocal laser scanning microscopy or in fluorescence microscopy. So if you just think about these three possible scenarios here, we have the wild type, for instance, in green, the FLE-C mutant in red, and the chemotaxis mutant in blue. If we now look at them in the fluorescent microscope, we can actually see that the bacteria are indeed fluorescing in different colors. And that makes a co-incubation, of course, very possible. And I have a little movie here. I hope the movie will start or not. It does not, right? Unfortunately. Let me see. No, cannot. Maybe I have to stop the pointer option. Aha, now you can watch the movie in action, and you can see that the green wild type is attracted to those red spots. The red spots are, show it again. The red spots are, that's the autofluorescence of the chloroplasts in the diatoms. And you hopefully can clearly see that the green ones are active, whereas the blue and the red ones, even so they are still motile, the chemotaxis mutants are motile, but I don't see a lot of chemotaxis mutants attaching here. So that's a nice documentation. And we, of course, also had different other options or different other approaches to show that there is attachment. In the Nature Microbiology paper from 2023, we have not investigated the interaction of Marinobacter adherens with diatoms, but of Marinobacter adherens with cyanobacteria. Now, cyanobacteria are termed pico phytoplankton because they are as sized or have a similar size as bacteria. They are bacteria, as a matter of fact, but they also conduct photosynthesis, and we use the same mutants and the same wild type in order to demonstrate that this exchange was reciprocal. So sugars given to the diatoms and amino acids given to the bacteria. In the Chemotaxis and Flagella mutant, these interactions were, of course, minimized. So the experiment which I would like to shortly talk about in detail with is the proteomics analysis. So the approach here is very simple. The approach is such that the two partners will be combined in the cell culture flask here, or the bacterium alone, the diatom alone, or the medium alone. And then a so-called attachment assay will be done after a certain incubation for 24 hours. We will, now I can put the pointer option again, it's easier to see. We take the mixture and we run them over plankton sieves, which have a pore size of 10 micrometers. These plankton sieves will allow free-living bacteria to pass through, whereas all bacteria attached to the diatoms, which have a size of roughly 10 micrometers, will stay attached to the diatoms or to aggregates, which of course need to be bigger than single diatoms. And then we will get a free fraction and an attached fraction. They will be separated from each other. As you can see, a lot of bacteria, of course, are still running through, so they are not attached. But then there's also a considerable amount of bacteria in the so-called attached fraction. So when we look at what is in the filter, and this is again a confocal laser scanning micrograph, we can see that these aggregates are amorphous. They are how can I say, they don't have a clear structure. They can have all kinds of dimensions. This is 10 micrometers here. So this particle here is much bigger than 10 micrometers. And the fluorescent signals are bacteria, which again are found inside the tap. The diatoms could not be visualized here, but it might also be that this particle here has only dead diatoms because we could not see the autofluorescence of the chloroplasts. So when we now, an alternative to this is that we investigate the attached fraction and the free living fraction with the help of Alcyon blue, because Alcyon blue is a dye which stains transparent exomolymeric particles. And when we look at the wild type, which now is supplied with a deep shifted red fluorescent protein, it's a variant of green fluorescent protein, and then visualize take the control, the Weiss-Flogy control. Here you can beautifully see the diatom cell. You can see the autofluorescence of the diatom cell, and you see, as with Alcyon blue stained, a lot of debris and a lot of exudate which the diatom is embedded in. Whether this diatom has produced all this or whether it's just a coincidence, we cannot tell, because we do not follow the procedure in a real-time scenario. We incubate for 24 hours and then we see what we harvest. What you can see now here in 3 examples, this is all the wild type, is that the bacteria that do not necessarily seem to be close to diatom cells, but that they are enriched in the transparent exomolymeric particles. And of course, you can from time to time see part of the autofluorescence of the diatom, like here and here. Eventually also here somewhere, but the majority of signal is, in this particular case, bacteria, which are residing inside the transparent exomolymeric particle. So bacteria mainly found inside the tap, and more inside the tap than, okay, sorry. Now I've shifted too fast. This is the previous slide, which is the delta Fli C mutant again marked with these DS red plasmid, and you can see that there's still attachment to the TAP, but lower density of the bacterium inside TAP than the wild type. And when we compare that with the GA mutant, we also have a lower density of the GA mutant inside the TAP as compared to the wild type. So that again, of course, proved what we had seen before, but it also enabled us to now take the biomass, the free living cells and the attached cells, and conduct proteomics with that. For this, we took the two fractions, we did microscopy and determined the CFU so that we know how many cells there are, and then protein extraction was conducted. In this case, because it's also a couple of years ago, we still did two-dimensional STS polyacrylamide gel electrophoresis. Today, status 2023, we would probably do a shotgun sequencing because we just got a new Orbitrap LC HPLC system. We then identify those proteins which are differentially expressed by comparing wild type and mutant, or wild type in the attached and in the free living, and so on. And we obtain, of course, best hits of strongly upregulated genes and gene products. We did this originally with the MALDI-TOF. Nowadays, we would do it with an Orbitrap mass spectrometer. So here are some of the results. And in order to guide you through the scheme, what the corresponding PhD student here has done is that we had, of course, the bacteria alone, and we had the bacteria and diatom together, separated into the attached ones and the free living ones. So those bacteria here in the so-called free living status are different from those bacteria because they have been exposed to the diatom. At least they had the chance to be exposed. And correspondingly, in the wild type, we saw aminobutyrate transaminase, glutamine synthase being upregulated in the attached fraction, whereas in the free living fraction, we have the flagellin upregulated, and we have other surface proteins and motility proteins upregulated. Also a TON-B dependent receptor, which of course again argues that these bacteria here need to take up sugars from somewhere, but we didn't find this TON-B dependent receptor down here. In the only bacteria fraction here, which is not a fraction, it's a separate sample, we just found general metabolism upregulated, and that probably has to do with the fact that we used a medium which is low in nutrients. If we now compare that to when we make a direct comparison of the wild type with the flagellin mutant, we did not find any differences between them in the attached fraction. However, we found in the free living fraction branch chain amino acid transporters upregulated, again, glutamate ammonia ligase and arginino succinate synthase. We cannot really make sense of those systems here, but these have been repeatedly found that the corresponding mutants need amino acids and need to uptake these amino acids because they are not supplied by amino acids by the diatoms. That's our interpretation. If we compare that with a GA mutant, the wild type, and the GA mutant, again, we didn't find any significant difference in the attached fraction. We also didn't find any significant differences in the bacteria alone, but we found stringent starvation protein, adenosyl homocysteine cystinase, and we also found the hydroxyl tetrahydrodepicolinate synthase. So it looks like the Chemotexis mutant cannot sense the signal anymore, which the diatom gives. If it were, then it would behave like the wild type in as much as that it would upregulate amino acid uptake systems. That is our interpretation. If we now compare the differences between the two mutants with a wild type in the attached one, then one can find amino acid synthesis and utilization as being upregulated. In the free fraction, motility and amino acid synthesis upregulated. And in the other two mutants, again, FLC amino acid synthesis utilization, and here utilization and starvation. That's the major difference. Yeah. Good. So, in other words, our heterotrophic bacterium doesn't seem to mess around with sugars. At least we did not find any massive hints for that. So in the conclusion and also in sort of like an outlook, the wild type of Marinobacter adherens colonizes transparent exopolymeric particle in higher density than the corresponding mutants. The proteins involved in amino acid metabolism are upregulated in the attached bacteria. Motility-associated proteins are upregulated in the free-living but diatom-exposed bacteria. If the flagellin mutant shows a higher expression of amino acid uptake than the wild type, and the GA shows signs of amino acid starvation without being able to upregulate it. It's just blind, so to say. Yeah, the blind cow, as we say in German. So there must be chemoattractants like amino acids present in TEP, and this is what we currently try to understand, which amino acids these are. There's a hint that it would be branched chain amino acids like isoleucine, valine, for example, and leucine. We want to quantify the amino acids in the TEP and in the TEP-free medium. And we want to create novel mutants deprived in amino acid uptake. And the last PhD student I had on this project was also successful with the mutant analysis. Now, unfortunately, she had to leave and we could not fully analyze these mutants. Good. With this, I would like to thank you for listening to this talk. And I, of course, also would like to thank my research team. There are a couple of Indian, like Neha Kumari here, for instance, and there was also Sujit, but he's not on there. But nevertheless, it's a very international team. We are from what's formerly known as Jacobs University, now Constructor University. We had to take a change in the name. The research was supported by the Deutsche Forschungsgemeinschaft, DFG, and these are the main PhD students who all contributed equally, more or less, to the entire marine project. Thank you very much for your attention. And now I would like to either stop sharing or I'm very open for any questions you have.
Very interesting lecture. A lot of new information. Very good. I like to know one thing, that is, what is the chemical nature of autofluorescence in diatom?
It's a chlorophyll. The chlorophyll itself autofluoresces when you excite it with the corresponding excitation wavelength, which is needed for the green fluorescent protein. Mm-hmm. The different variants of green fluorescent protein are all excited at the same wavelengths, but emit at different wavelengths. And unfortunately, the emission spectra for these deep shifted red and yellow fluorescent and red fluorescent are very close to the original one. They require a very narrow bandwidth for the fluorescence microscopy. And chlorophyll, I'm not sure whether it's only chlorophyll or whether it's also something else, unfortunately emits with this red, this clear red signal as well.
And what is the nature of chlorophyll?
What is the nature of chlorophyll? It consists of the phytol ring and the manganese containing. I don't know how to explain that, but you should know.
The other, I like to know that when you have the A-flagellate butane you have made, how do they get attached to the diatoms?
Yeah, well, that's what we argue, that the flagellum is very important for the initial interaction. So from biofilm research, we know that flagella are very often the initial, and also pili are often the initial sites for getting an interaction between a surface, any kind of larger surface, and the bacterial cell. So at the moment, our best guess is that the flagellum, this polar flagellum, is, for instance, aligning with the surface of the diatom and is allowing the bacterium to stay in close proximity. Which other factors then contribute, whether they are surface proteins which contribute to this, we would only find out if we do a random and non-biased approach. All our approaches were biased. They were based on on a hypothesis that flagella are important, chemotexis is important. We also tested a couple of other things. Pili, for instance, do not seem to be important. I didn't show the results here, but we had a pilus mutant, PIL-A, and that mutant did not differ from the wild type at all. But a random approach, a black box approach, watching at what mutations would lead to a lack of interaction, would need a very smart screen. What we did was we did IVIT screen. I don't know whether you've ever heard it, in vitro expression technology screen. And that is a modern technique which researchers on medically important bacteria have conducted. It's similar to signature tech mutagenesis. These both methods are interchangeably. And with this, you make a random approach. So you look what genes are upregulated when the bacteria are in close proximity. And then you find again amino acid metabolism.
Mm-hmm. Right.
So how they physically interact, I do not know. Maybe the interaction is not very tight.
But it's research.
Yeah, well, they could eventually even randomly stay in the transparent exopolymeric particle because it's a gluey stuff. So that's quite possible.
If I understood you correctly, the organic carbon is produced by interaction of bacteria and diatom, and then they go down to the bottom of the sea, right?
The organic carbon is produced by photosynthesis.
Yeah.
And
they come up to the CO2 level from the bottom to up. What is the driving force?
I'm not sure whether I understand your question. Like the carbon, the organic carbon is made by photosynthesis. That's
right.
And then
it's down in the bottom of the sea.
Yeah.
Right.
Yes.
And they degrade into CO2, inorganic CO2.
Yeah, because of there are again heterotrophic microorganisms also in the deep sea, which of course also consume the organic carbon and make CO2. However, there's a lot less and much slower process there. The temperature very low, the pressure very high. They need to be, most of the organisms down there are chemosynthetic, lithotrophic. So if there are heterotrophs, which I have no doubt that there will be heterotrophs there as well, they will be extremely slow in metabolizing. Whereas in the light flooded zone of the ocean, we have reasonable temperatures, and consequently there will be a lot of metabolism there. That's why the majority of carbon dioxide, which gets recycled into the atmosphere, is happening in the photic zone.
Right. Very good. Very good. Perhaps many of you do not know that one of our students, they went to his laboratory to do PhD, and he did very well. He was coming from Allahabad, from economically lower class, but he was so keen to do PhD from Germany, and he was accepted by him. He likes Indian students because they are bright.
You are talking about Abhishek, right?
Yes.
Yes.
And he's in Austria now.
Yes, I think so. Yeah.
Yeah, in Austria now. And I suggest that many of our MSc students who complete MSc this year read this nature paper and come out with a novel idea to do further research, and he'll be very happy to accept you for doing a PhD in his laboratory. Your laboratory is very good. I have been so many times, and try to think of this nature paper, read that paper, then come out with some novel ideas, and give your ideas to the professor, and he may invite you for doing PhD over there. Thank you very much. Thank you. Yes.
Thank you so much.
I'll
Thank
ask
you.
faculty student to ask questions.
Hello. Hello.
It's okay. Yeah.
Hello, sir. This is Dr. Naveen Joshi from AIMT. My question is, first of all, thank you for sharing such a great knowledge here. And my question is about methodology. How you created these bacterial mutant? It was site-directed mutagenesis you did? Or something?
Yes. Yeah, so we did site-directed mutagenesis using homologous recombination. Yeah, I know that nowadays you would do everything with CRISPR-Cas, but at that time we cloned by PCR the flanking sequences and part of the coding sequence of the gene of interest. We cloned them into a normal vector in E. coli, then placed an antibiotic resistance gene cassette in between, and allowed for homologous recombination by introducing the plasmid, which now acts as a suicide plasmid, into the Marinobacter adherens, and then select for the antibiotic resistance for a homologous recombination. We have to then prove by PCR that the homologous recombination has happened, and we could show that. We also sequenced the corresponding region, so we demonstrated that the resistance cassette was in there. As a matter of fact, at the Nature paper, they then ask us, the reviewers ask us, could you complement the mutant? And complementing a mutant from a gene, or complementing a gene which is located in a 32 kb operon, chemotaxis and flagella biosynthesis are both in operons, is impossible. We would have to complement with the entire operon, because you cannot just simply complement with one single gene. The operon is destroyed, of course. You know? Now, in the case of the Fle-C mutant, that we could still see the hook is contributed to the fact that the hook gene is not in the same operon. Otherwise, we wouldn't also have knocked out the hook gene. So, yeah, this is the technology. It very much depends also, of course, on luck, because this homologous recombination, after all, is a random event. You cannot force it. But when you conduct everything from the genetics point of view accurately, you can generate any kind of mutant.
Thank you.
Okay. Thank you very much for the nice questions. Highly appreciate it.
Now, I would like to invite Professor Dr. Rajesh Mishra for his theme lecture on the stability and folding of bacterial alpha amylases. Professor Dr. Mishra is currently a professor in the School of Biotechnology, JNU, New Delhi. He has completed his PhD from the Center for Biotechnology, JNU, and the Department of Physical Biochemistry, University of Potsdam, Germany. His research area is protein science, folding aggregation, and amyloid fibril formation. He has been awarded DARD long-term fellowship under the Sandwich PhD program at the University of Potsdam, Germany, graduate teaching assistantship at the Department of Biochemistry, GB Pant University of Agriculture and Technology. He has published many research papers and articles in the journals of repute. A very warm welcome to you, sir.
So thank you, Professor Verma, for giving me opportunity to speak in a department. Although I'm not new, I'm familiar and a member of the DRC, but it's the first time I'm going to speak. One of the reason is because my area of research and matching with the objectives of your department. So I chosen to that topic, which is stability and folding of bacterial alpha amylases. So briefly, first I will introduce what my area of research is, and then after a couple of slides, I'm coming to the topic of my talk. The proteins are synthesized on the ribosomes, and it starts. I will start with the cellular protein synthesis. It starts with on the ribosome, and the newly synthesized polypeptide. Either it may go straight to form the native state through the intermediate state, or it can go to the inclusion bodies. Sometimes many of you have observed during the the bacterial protein expression in E. coli, where your desired proteins from the inclusion bodies, and also these intermediate states, they can form the another type of protein aggregates, the ordered aggregates, which is commonly found different disease conditions, and this is known as these order aggregates are known as amyloid, and these amyloids are responsible for more than 30 human diseases, and also in the animals these causes. The characteristic feature of these amyloids are they are cross beta sheet rich structure. Now the folding is assisted by the molecular chaperones, and also after the protein folding, post-translational modification takes place. So in our laboratory, we are focusing on two aspects. The first one is the protein aggregation and amyloid fibril formation. We're using a number of proteins as a model system, like type 2 diabetes related amyloid, where islet amyloid, polypeptide, IAPP, and insulin is involved. Model proteins like alpha lactalbumin. We are also working whereas there is a thin line between protein aggregates and amyloid is a human gamma D crystalline. And we have just initiated a new project on the functional amyloid of a bacterial curly protein, where the major protein involved is CSGA. The second aspect of our research is a protein stability and folding, and where the current talk I'm going to give you about the stability and folding of proteins, particularly thermophilic-like proteins. And also we are working on the acid stress chaperone in E. coli, HDEA, HDEP. Now coming back to the topic and the enzyme alpha amylase, which is one of the very, very industrially important enzyme, and it catalyzes the hydrolysis of internal alpha-1,4 glycosidic linkage in starch and other polysaccharides. It's a ubiquitous in nature, and one of the interesting thing about it's a wide the adaptability, the temperature adaptability, right from the the less than 20 degree to more than 100 degree, and the pH it can vary from 1 to 11.5. Now this is one of the very, very important, industrially important enzyme, and a lot of biotechnology companies they are producing, and its application includes from the detergent to the textile industry, to the baking, to the paper industries, and so forth. So industrially, it's still a very, very important enzyme. Now, it's not only industrially important enzyme, it's present in the diverse form of life, right from the plant, human, plant, animals, and microbes. But the industrially important enzymes comes from the bacteria or fungi. And depending on their ecological adaptation, the alpha amylase producing organism, they could be thermophile, they could be mesophilic, psychrophilic, barophilic. Barophilic is the term which is used where the organism go under high pressure, like a deep ocean where the pressure is more than 1,000 bar. It's a special kind of adaptation. Acidophilic, halophilic, and alkalophilic. So bacterial and fungal alpha amylases are the most important. Now the most of the published work is focused on the application, the identification of new enzymes and their application. And not so much focus has been done on the structural aspect. And it's important to understand the structure before we can fully understand its function. So let us briefly look at what is the structural diversity in bacterial alpha amylases. So here I am presenting a different, the crystal structure of different classes of bacteria. So for example, here you have a psychrophilic, then you have a thermophilic, hyperthermophilic, halophilic, acidophilic, and alkalophilic. You see the common thing, they all have 3 domains. They are represented in 3 different colors, except the one where you have an additional N domain. So more or less, they look quite similar, and most of them having a metal binding site and the catalytic site, they are quite evolutionary conserved. So apparently, they don't look they're much different from just looking the structure, but they have a different adaptability, and this adaptability can be seen by looking at just as the temperature optima. So for example, psychrophilic bacteria, which is one of the most studied psychrophilic bacteria, is Pseudo-Ultramonas haloplanctis, where temperature optima is 4 to 25 degrees centigrade, while the Bacillus licheniformis or the Bacillus stereothermophilus is a 90 degree and 70 degree. And in the extreme cases, in the hyperthermophilic, there are more than 100 degree temperature optima. So why there is so much of variation? For this, a lot of effort is going on to understand the basis of the adaptability, is temperature adaptability. That's why some bacteria is thriving at 4 degree, 4 to 20 degree, and why some bacteria is thriving as a more than 100 degree. But to understand that, you have to understand the factors which affects the stability of a proteins. So stability of a proteins is affected by the mostly non-covalent interactions. And these non-covalent interactions, this is particularly from the pedagogic point of view for the students, so you can get a little more attention that what is the basis of a protein stability, and that is the non-covalent interactions. So ionic interactions, hydrophobic interactions, and hydrogen bonding. Of course, the van der Waals interactions is present between any two atoms. And these non-covalent interactions are determined by the side chains of amino acids. So that you call it, you have a charged residue, you have a proline, you have a glycine, and so forth. One covalent interaction which is involved in the stability of a proteins is the disulfide bond, where the cysteine is involved. So that's only covalent interaction which is involved in the stability of the proteins. So it's obvious that first you compare the amino acid sequence from the different classes of a bacteria: saccharophilic, thermophilic, mesophilic, acidophilic, and alkalophilic. So I'm not going to just tell you everything, but just to highlighting that if you look at some of the amino acids which are important for the stability of a proteins. So ionic interactions are obviously, they are mainly controlled by the charged residues like positively charged lysine, arginine, negatively charged aspartic acid, and glutamic acid. Then you have a hydrophobic interactions, the hydrophobic residues, and then hydrogen bonding like a polar residue, serine, and so forth. So here, but there is a role of one other residue which is very important in the stability of a protein, that is a proline. And it is related with the thermodynamic stability because because of this the entropy decreases. So if you look at the saccharophilic bacteria, they have a proline content is quite low, while in thermophilic bacteria and hypothermophilic bacteria, proline content is higher. And so if you look at the charged residue, which is the lysine, arginine, positively charged, aspartate, and glutamate, negatively charged. In saccharophilic, the content is lower, and in the hypothermophilic, thermophilic, they have the higher content of a charged residue. And what is the relationship of this with the stability that I explained you? That it increases, if you have a charged residue more, there will be possibility of a more ionic interactions. And not only the ionic interactions, but it will form the cluster of ionic interactions. And then the covalent bond, disulfide bond, which increases the stability. You see, the nature has compensated saccharophilic bacteria by adding some amount of cysteine, which provides the stability. So for our work, we have consciously chosen two bacterial alpha amylases. One is obtained from the Bacillus amyloliquefaciens, which I am abbreviating BAA, and another one alpha amylase is obtained from the Bacillus licheniformis, BLA. Although both are mesophilic in origin, but BLA behaves as a thermophilic while BA as a mesophilic. And why BLA behaves as thermophilic and BA mesophilic? Because if you look at the TM of a BLA, which is a Bacillus licheniformis, it has 101 degree, and BA has 86 degree. Interesting thing about these two enzymes are that BLA and BA share 88% sequence similarity and 81% sequence identity. But in spite of so much of sequence similarity and identity, the TM is significantly differ, which is a 15 degree. Structure wise also, they look quite similar with the three, domain A, B, and C. Both proteins have calcium bonds, which requires for its stability and the function. So then why there is so much of difference in thermal stability when the proteins are so similar? And if you look, compare the amino acid sequence of both proteins, so you can look at even the positively charged residues are almost similar here in the case of thermophilic, like alpha amylase, BLA is a 49, here you have a 50. So the negatively charged is not much difference. Proline, which is the important factor for protein stability, is also similar, and there is no cysteine. However, there is one significant difference between these two alpha amylases. One is the presence of a number of histidine residues. So if one would look for the stability, the first thing is to compare the amino acid sequence. So here the histidine, there are 24 histidine in the case of a thermophilic alpha amylase, and there's only 14 histidine in the case of a mesophilic alpha amylase. Now, this information, which is abundantly present in a lot of sequences known, so one can predict that yes, this protein will be more stable by just comparing the amino acid sequence. But problem is here, by just looking at the amino acid sequence, you cannot really tell what is the basis of the stability. So structure and sequence comparison does not provide the complete picture. And it's actually the answer lies in the dynamic nature of the protein. And that's where our work I'm going to talk about, our results. So most of the work earlier on these enzymes related with the stability and folding is confined to the thermal stability. And to understand the origin of this stability, we have to go for not only thermal, we have to go for the chemical induced unfolding like guanidinium chloride and urea, which are known protein denaturant. And the outcome of this study will provide the molecular determinants of protein stability, which may useful for the designing you can mutate the alpha amylases to make it more stable under different harsh conditions. So what we did study the protein unfolding of these both enzymes, BLA and BA. And what's a probe? How we studied the protein unfolding? We studied taken as a tryptophan as a probe. Tryptophan is a amino acid residue, and this has an intrinsic fluorescence property. So tryptophan is not only useful for the taking the absorbance, determining protein concentration, but it is also intrinsic fluorescence. So both enzyme has a 17 tryptophan, so that's a fantastic. That gives the good opportunity for us to use at molecular probes. And if you look at these two spectra, the black and the red. So black one is the folded state and the red one is the unfolded state. So what's the difference you can see? The difference is a twofold. One is the intensity, fluorescence intensity. In the unfolded state, fluorescence intensity decreases. But the more important thing is the wavelength shift. So that is very important parameter to understand protein stability. Similarly, in the case of a mesophilic alpha amylases, BA, you have a decrease in fluorescence intensity as well as the wavelength shift in the unfolded state. Using this probe, we did a series of experiments. So look at these figures, which is the experiments of equilibrium unfolding of BLA and BA in GDMCl and urea. GDMCl guanidinium chloride, this is a charged denaturant, and urea, which is the uncharged, but both denature the protein. Of course, if it's uncharged, it has a different mechanism. When it's a charge, it has a different mechanism. So that's why we have chosen this both denaturant, charged as well as the uncharged. And rationale is that because stability is provided by the one of the important contributing factor is the electrostatic interaction. So how the electrostatic interactions are affecting the stability of the protein. So if you look at the figure A and B is the BLA and B is a BA. So, and these are, you don't go with the different graphs, but just you can look at the BLA is more stable in presence of guanidinium chloride compared to BA. And how you monitor? On the x-axis you have a guanidinium chloride, and y-axis you have a wavelength shift. Wavelength shift, what we have obtained from the tryptophan, intrinsic tryptophan fluorescence. So protein unfolds, here the wavelength shift, wavelength maxima increases from 340 to 356 and so on. So thermophilic protein enzyme BLA is requires more guanidinium concentration than the mesophilic. That means this is a more stable than the BA. Now, what is the effect in the presence of a urea? So urea is uncharged. So when we look at the left side, see, thermophilic enzyme BLA does not unfold in the presence of urea, while BA unfolds. That means it's a much stronger than the BA. Thermophilic enzyme is much more stronger than the BA. Now, comparing the unfolding parameters like how much concentration CM, like the TM is a melting temperature where half of the protein is denatured. TM in DNA, all of you are aware, but TM in protein is also the temperature at which half of the protein is denatured. So like TM, here we are talking about the CM, concentration of a denaturant, which requires half of the protein to get denatured. So if you look at the BLA, CM is 1.3 molar for the guanidinium chloride, while in the case of a BA, it is just 0.62 molar. In the case of urea, BLA could not be unfolded, while the BA requires a much higher concentration, 3.4 molar. So BLA has a higher value of a CM during unfolding in GDM chloride than BA 1.3 molar, and BLA remains folded in urea. You cannot unfold at pH 7 even after 6 days. So this was intriguing, that why BLA does not unfold even if you keep it for the 6 days. So what we did to unfold the BLA, we lowered the pH. When we lower the pH, what it's going to do, it's going to alter the hydrogen bonding. It will disrupt the hydrogen bonding and the protein should unfold. So if you look at the pH 3 and the another set of experiment at pH 7, we added 1 molar sodium chloride. So effect of acidic pH and ionic strength is provided keeping the pH same, but by adding 1 molar sodium chloride in urea. So we wanted to unfold the protein, we just in presence of urea, but now we are adding the parameters, lowering the pH and adding the sodium chloride. So what is the effect on this? You see that for unfolding at pH 3, BLA was incubated at various urea concentration. You see on x-axis, even at 8 molar, you can see up to 8 molar. And unfolding in varying concentration of urea, where 1 molar sodium chloride was added. The idea was that if the salt is the factor in guanidinium chloride, that is supplemented by the sodium chloride. So BLA remains folded in urea even in the presence of 1 molar sodium chloride at pH 7, while in the acidic pH it unfolds, while unfolds rapidly in urea at pH 3, which indicates the role of ionic interactions. Because if you lower the pH, then it starts unfolding. That means the ionic interactions are playing an important role. After the unfolding, we wanted to correlate the structure-activity relationship, because an enzyme is active when it will show the its active enzyme will be the native state when it will show the activity. And so we have chosen 3 probes. One is the activity assay. So protein will be in the native state, it will show enzyme activity. Then we also looked at the secondary structure, and that we monitored by the circular dichorism, particularly the alpha helical structure, and which we monitor at a particular wavelength 222 nanometer. And then a global tertiary structure we monitor by the tryptophan location by the fluorescence spectroscopy. So if you look at the BLA and BA in the presence of guanidinium chloride, you see the red is the fluorescence, black is the fluorescence, and red is CD, and blue is the activity. These 3 different colors. And BLA, you see all these 3 probes do not overlap. That means the loss of a tertiary structure, secondary structure is not simultaneous. While in the case of a BA, they are overlap. That means both secondary and tertiary structure is losing simultaneously. And this is very important in the case of a protein folding, unfolding, because that shows the hysteresis. That means the unfolding pathways are different. So to understand why this is a non-coincidental unfolding in BLA. Of course, in the case of BLA and BA in presence of urea, I mean, we know that it doesn't unfold in the BLA, doesn't unfold, and BA it does unfold, and here we see the overlapping. So in the case of BA, we saw both in the guanidinium chloride and in the case of urea, there is an overlap, but in BLA, there is a non-overlap. And this is a clear indication that they must be having the different unfolding pathway. So if they have a different unfolding pathway, that means they are forming the different kinds of a structure. And to probe the different kind of a structure, we use the another probe, which is the ANS. ANS is 1-anilonaphthalene 8-sulfonate. This is the extrinsic fluorophore. Like a tryptophan is the intrinsic, ANS is the extrinsic. So where there is a hydrophobic patches, this extrinsic fluorophore binds and the intensity increases. So here you see in the figure A, in the case of a BLA, if you unfold, as we unfold the protein, more hydrophobic patches, more intensity increase, as well as the wavelength shift. While in the case of a BA, there is a not so much difference. The two differential unfolding probed by ANS indicates that the intermediate or molten globule states which are formed during protein unfolding are excuse me, different. So again, we tried the same experiment in the presence of urea, both BLA and BA, and again we saw the differential pattern. So the different unfolding pathways of BLA and BA in guanidinium chloride, and the same different unfolding pathway of BA in GDMCl and urea. So why this different unfolding pathway, and what is the relationship of this differential unfolding pathway in the stability? So does this difference reflects in the kinetics of unfolding? Kinetics, we follow the reaction over a period of a time, and here we can see when we follow the unfolding kinetics, the same reaction continuously over a different time periods. You see on the x-axis you have a minutes, and we can see clearly there are two phases, both BLA and BA, but they are rate constants. So BA, it folds faster, and BLA, it folds slower. So the rate constants are different. And this biphasic unfolding, this indicates that what we have saw this three domain, so the simultaneously unfolding of two different types of a domain. And then we also calculated the chevron plot of BLA and BA and guanidinium chloride, which shows the linear dependence of a logarithmic value of unfolding rate constant. So unfolding rate constant, basically we calculate from here whether the folding is a faster or slower. So slower unfolding rate, which is present in thermophilic protein. This is a very important parameter, which has been least explored in the case of thermophilic proteins. If the rate of unfolding is slow, that means protein will be more stable. So even if you know the sequence and the structure, but without understanding the unfolding kinetics, it's a difficult to predict. And it's a very important tool. If you study the unfolding and you calculate the unfolding rate constant, then you can predict that which protein will be more stable. So slower unfolding rate constants of BLA indicates the kinetic origin of the stability. That's what we showed here. So if I conclude this part of this, the work, that both BLA and BA shows cooperative unfolding transition. BLA shows more resistant in GDMCl guanidinium chloride than urea. BLA exhibits extreme stability in urea at pH 7. So even at 6 days, it doesn't unfold. While BA shows cooperative unfolding in urea with CM higher than GDMCl. Obviously, urea requires a higher concentration than guanidinium chloride, which might originate due to hydrophobic protein core and strategic salt bridges in BLA. So structure function relationship and ANS binding studies have shown that partially unfolded intermediates were populated during the unfolding of a BLA in guanidinium chloride, and both BLA and BA exhibits the biphasic kinetic unfolding transition in guanidinium chloride, where BLA has a slower rate of unfold. So this rate of unfolding is an important criteria to understand the stability of the protein. So BLA has a slower rate of unfolding in guanidinium chloride than BA, which indicates the kinetic stability of BLA than the BA. Now, the smaller part I'm going to talk about just briefly, that this was the reflection in the unfolding pattern. Is there effect on the refolding? Refolding means you unfold the protein and then you refold again in the refolding buffer. So what we have done here, we have used the probes, the stabilizers, so which are known to stabilize the enzymes, which they are very useful in the industry. So the polyols like ethylene glycol, glycerol, erythritol, and xylitol and sorbitol, these are the polyols which are often used to stabilize the protein, and the sugars like sucrose and trehalose. So what will be their effect in the refolding? Are these stabilizers have the same effect? Thermophilic is more stable, mesophilic is less stable. Is the nature has optimized thermophilic that you cannot enhance its stability, or you can enhance the stability? So that question we asked, and we just this the schematic of how the refolding is done. So if you unfold alpha amylase in 6 molar guanidinium chloride, you dilute it, and then for vortex for the proper mixing, and then sample incubate for the so that refolding is complete, and then you monitor by the enzyme assay, circular dichorism, and fluorescence. So what we saw that this three the co-solvents, sorbitol, sucrose, and trehalose, they are the most useful in the in the BAA refolding. So if you the black is a BLA and the gray is the BA. BA is a mesophilic. So you see the thermophilic protein refolding is not really much assisted by these folding enhancers, stabilizers. While you see in the gray where the refolding yield is increases, and this is the mesophilic. That means the mesophilic protein can be enhanced, their stability can be enhanced, their refolding can be enhanced. So sorbitol, sucrose, and trehalose, the trend is same in all three cases. BA shows more enhancement in the refolding than the BLA. And if you draw a pattern that how the number of hydroxyl groups that I talked about here, that from different molecules, ethylene glycol, for example, has a 2 hydroxyl group, glycerol has a 3, erythritol has a 4, xylitol has a 5, sorbitol has a 6. So how they affect? So if you can see here, the x-axis is the number of hydroxyl groups. So black is the BLA and gray is the BA. So refolding is enhanced as we increase the number of hydroxyl groups, and this is a more effective in the case of a mesophilic protein, mesophilic enzyme BA than the BLA. Then we ask the question whether this is does it affecting the aggregation as well? So because if protein is 60% refolding, what is happening to the rest of the protein? That means it is going to the in the aggregated state. And what we can see that see the black traces is these are the control, and the red and the green traces, these are presence of a different co-solvents. So left side you have a thermophilic BA, and the right side you have a BA, mesophilic enzyme alpha amylase BA. So what we can see that all the co-solvent reduces the extent of aggregation. So they are preventing to form the protein aggregates during refolding of both BLA and BA. But this effect is greater for the BA mesophilic, where we have seen that this co-solvent enhances the refolding. So the same co-solvents also reduces the aggregation. So the nature has, it means the optimized, that where they are less stable, they can be further stabilized. Where these are optimally stable, they can be less stabilized. And to understand the mechanism that how these co-solvents are helping in the refolding. So here we see we studied the refolding kinetics of BA because that's where we have seen the effect. And then we added the co-solvent at different time points. So trehalose was taken. So we can see when there is when the co-solvent present from the beginning, that means excuse me, 0 time, then it is effective. But when you add after the 0.5 minute, 1 minute, and so forth, there is a no effect. So no increased refolding of BA was observed upon delaying addition of a trehalose even by 30 seconds. So stabilizers has to be present in the beginning. This co-solvents required at very early stages of refolding, and this indicates the fast folding nature of a BA. That means BA refold very fast, and that's why even you add after 30 seconds, it doesn't help. Then we also followed the same BA refolding by the far UV-CD spectroscopy, which is the indicator of the secondary structure. So if we can see that after 30 seconds, refolding in buffer where there is a no additive, it's almost no structure, but in the presence of a trehalose, it regains almost 40% of the native structure. So low gain ellipticity at 222 nanometer, which is the indicator of the secondary structure, again points towards the role of a trehalose at early stages of a refolding. That means you have to have a stabilizer which can stabilize your refolding intermediates at the early stage. To further understand its mechanism, we added the urea effect of urea on refolding of a BLA and BA, and then what we saw, because if the during refolding these small intermediate states can easily be denatured by addition of urea. So low urea concentration exerted positive effect on refolding of a BA, while low urea concentration showed negative effect on BLA refolding. That again shows that the nature of refolding excuse me, of both BLA and BA is different. Excuse me. So this is probably the last slide I'll have. And to further understand what when we destabilize by urea, then we stabilize by this co-solvents, and we have taken the trehalose. And what you can see that glycerol alone, for example, 32% refolding in BLA, 54% in BA, 1 molar trehalose 32, 67, sorbitol 37 and 56. But when you add 2 molar urea plus 6 molar glycerol, here it's 23% in thermophilic protein, and here is a 42%. Here you have a 14%, here you have a 47%. In thermophilic, here 6%, and then 46%. That means the stabilizers works better in the case of mesophilic proteins. So in that way, we delineated that how the refolding pathway, the intermediates of refolding can be destabilized by urea and stabilized by the trehalose. And this is the clear direct evidence that if you have the mixture of a stabilizer and destabilizer, we could see that mesophilic enzyme BA is more affected, and that again point out that in spite of a sequence similarity, they are different in their unfolding and refolding pathway. So to summarize this part, the trehalose, glycerol, and sorbitol found to be the most effective for refolding of a BA, well, no co-solvent seems effective for refolding of a BLA. BLA is more aggregation prone, so more stable, more aggregation prone than BA. Refolding kinetics of BA and trehalose showed that co-solvents are required at early stages of a BA refolding, and differential behavior of a BLA and BA towards urea during refolding indicates that BLA and BA may fold via different refolding pathway. So this was one of the early project I got from the UGC and the facility DBT Builder facility, and most of the work presented here is my PhD student Aziz Ahmed's work. And thank you all for listening. I've tried to explain. If you have any question, please ask me.
I see that you have got gold mine in hand. Why not you transfer your knowledge and wisdom to some industry?
Aziz Ahmed got just a postdoctoral position in the University of Minnesota Pharmacy Department, which is one of the topmost lab in the world. They are working on protein formulation. So this co-solvent effect, how to stabilize this therapeutically important proteins. In fact, we are working for quite a long time, and now we have enough knowledge,
Yes.
but it's still not moving forward. But thank you very much. I think that's a time that we should go
move forward with industry.
Yeah.
Now, why I asked this question? Because some years ago, my daughter had come from North Carolina, and she visited a company near Jaipur, where they're taking the textile waste cuttings from tailors and converting them into paper. And they say it almost 30 days to convert into paper form. So why not you apply this knowledge there, and they can move faster in a week time instead of 30 days.
Thank you, sir. We will
this
try.
has a great industrial importance.
Thank you, sir. It's a very encouraging suggestion, and I will try.
Thank you. Thank you. Yeah. Well, do you want to ask something? Go ahead.
Hello. That was me. Yeah. Nice presentation, sir. Excellent piece of work. My question is a bit general, actually, regarding the protein folding.
Yeah.
So we know that when protein is misfolded or somehow wrongly folded, then they can cause some diseases, like we have seen in prion-related diseases, the
Yes.
Alzheimer's, and lot of other diseases. Like, so the question is, do cell have some way to detect that this particular protein is wrongly folded or misfolded? Or when it is detected, then how the cell will regulate its degradation and whatever required?
Well, the cell has its own system, and that's why all of us are not affected by this misfolding and diseases. So protein degradation machinery, proteasome system, normally when the protein misfolded, they are recognized by the proteasome machinery and they are degraded. So they are not able to accumulate. But under some circumstances, it is not recognized, or sometimes you have the mutant, which is not recognized. Then they start depositing as a protein aggregates. But once it starts, then these aggregates cannot be cleared or degraded, and they deposited in the brain. So we know the Alzheimer's, Parkinson's are because of this. Prion protein is the classic case where the protein misfolding occurs. These aggregates are extremely stable. Even 6 molar guanidinium chloride, 8 molar urea, you can't unfold these protein aggregates. But yes, at the early stages, most of the time they are detected, and the partially folded or unfolded protein goes through the proteasome machinery.
Thank you, sir.
Yes, sir, I know. And also the Pantnagar product, biochemistry.
Dr. Rajesh, very good presentation. I have a basic, for my knowledge itself, I have a great question. You have said that 101 is thermophilic and 86 is mesophilic. Does it fits the criteria of the mesophilic?
No. It does not fit in the classical, so it's difficult to argue with the trained microbiologist. But for us, for a chemist point of view, there is a difference of a 15-degree TM with the 88% sequence similarity. So 15 degree is too much when you have 88% similarity. So, and actually both proteins are, both organisms are mesophilic in origin. Third thing, when the calcium is present, then they show this 101 and the 86. But when the calcium is removed, their stability is decreased to the 20 degree and so forth.
So that might be the calcium is present at the active site, which is enhanced.
Yes, so not active site actually is a nearby pocket which enhances the stability. So when you remove the calcium, excuse me, then the stability lowered. So from not in true sense, but yes, from our study point of view, this is a model for the thermophilic protein, and this is a model for the mesophilic protein.
So does the calcium is present in the vicinity of both the enzymes?
Yes. And actually removal of calcium leads to the exactly the same, I think it's a 25 degree centigrade drop in the TM. So the role of calcium actually nullified in the both cases. What was the striking here is the 24 histidine and the 14 histidine.
That's my next question.
Which is the new, that's why we got interested. And that's why, because sequence comparison only will tell you that this is a 24 histidine and this has a 14 residue, 14 histidine. But that should reflect in the unfolding kinetics. The unfolding rate should be lower in the case of a thermophilic. It should unfold with great difficulty compared to the mesophilic protein. And that's what we have shown, that the BLA unfolds slower than the BA.
Does the histidine has the folding and role in the folding and unfolding of the protein?
Well,
actually, normally cysteine is reported as a folding and unfolding. Histidine has a
no, histidine is actually here playing a role in this. We have not shown any the direct evidence. Obvious would be do the mutagenesis experiments and probe the role. But yes, it does affect the not the charge nature, but hydrophobicity and the location of a certain histidine in BLA, thermophilic protein, plays a very important role. So that is one of the reasons why BLA is more stable than the BA. So histidine is a key player. Otherwise, for the normal knowledge of especially the students, you should always look the amino acid sequence, and the proline and glycine, the two are important residues which one should look for, whether the protein will be stable or not, whether it will be flexible or not. But here it was the histidine, and that's what we got interested, that what histidine is doing.
One another basic questions. As you have shown that different microorganisms which has a category of the acidophilic, thermophilic and mesophilic, but you are finding the thermophilic enzyme from a mesophilic strain. Does the enzyme produced by the thermophilic organism and this enzyme which is produced by the mesophilic organism, although showing the thermophilic properties, does there is any similarity? It's not your work at present, I know.
No,
but
see, essentially it is only mesophilic in origin, but it has all the traits of a thermophilic enzyme. And obviously thermophilic, hyperthermophilic like pyrococcus and so, which are hyperthermophilic, they are even more stable, more than 100 degree. And there, actually this was the review we published, because there are a lot of reviews, we write very less, but if you just, I'll show you here.
that is
Yeah.
not this, before that, I think. Yes, this one. Temperature optimized even over there.
So this hypothermophilic, where you have 100 and more than 100. And if you look at one interesting thing, we found that normally cysteines or disulfide bonds are not present in thermophilic, hypothermophilic. But here the completely different. Psychrophilic also have cysteine, and here the hyperthermophilic also have a cysteine. So disulfide bond is probably the complementing way to enhance the stability in the case of thermophilic proteins. And also you look at the charged residues here. It's a known fact. So it is not only the positive and negative charged, it is the cluster of charges that are responsible for the enhanced stability in thermophilic proteins. Although there may not be much difference in the number of charged residues between thermophilic and mesophilic, but their location, they are clustered around, and that enhances the stability much more than the individual pair of charges.
Thank you, Dr. Rajesh. Anybody else?
Find the replacement of don't survive because of COVID.
No. I think the position will be advertised maybe soon. And he was working on the malaria biology, and earlier he was trained with the Professor V.S. Chauhan at ICGB. So hopefully somebody will join the department.
Anyone, any question, please?
Some of the things for the students, many of you appear in the CSIR, UGC. These methods or techniques are very important. So even if you have not listened the lecture carefully, or you could not follow, please go and read these methods, which are very useful for your different exams and different entrance exams or anywhere for the research. For example, fluorescence spectroscopy, CD, not the pure physical chemist point of view, but from the biochemist point of view, how fluorescence spectroscopy can be used. And here I showed you the tool, simple tryptophan can be used to study the protein conformation, intrinsic fluorophore, like CD, circular diaquarism, how 220 nanometer signature peak can be used to follow
Dr.
the
Rajesh, in general, microbiologists are a little bit apprehended towards the biochemistry. So that's true also.
No, but at this stage, there is no demarcation. Eventually, this is also microbiology, this is also protein chemistry, and this is also the spectroscopy. So they are much the young, and they're in much more modern era where they should.
You press the button which is they are lacking, I think.
So one of the purpose of my visiting here to just stimulate, ignite the students so they could get interested into asking these fundamental questions in micro.
One student.
Good afternoon, sir.
Yeah.
Sir, you talked about the diversity of alpha amylases, and in that you mentioned cycrophilic and barophilic.
Yeah.
Sir, they are both dependent on the criteria of pressure. So, what's the major difference between them?
No, cycrophilic is where they grow optimum at a lower temperature, and the barophilic, their adaptability is the temperature might be lower, but their adaptability, they survive in the like a deep sea under high pressure. So normal atmospheric pressure is the 1 bar, and if you go to the deep ocean, there the pressure is 1,000 bar. So imagine that how a protein, a microorganism's protein is surviving under the 1,000 bar. So what kind of adaptation they have? So that's where they are the different cycrophilic and the barophilic. But that's an actually very important area of research to understand the deep oceans microorganism, their adaptability, their enzymes and their proteins.
Thanks.
Thank you.
Anybody else?
Okay, then thank you all
Thank
of you,
Dr. Rajesh.
for
Thank you.
listening.
Thank you, sir, for the thought-provoking presentation. Now, I would like to invite Dr. Monica Gupta for the vote of thanks.
Thank you, Ayushi. On behalf of AIMT family, I would like to express my sincere thanks to Honorable Founder President Sir, Vice Chancellor Madam, Chancellor Sir, Dr. W. Selvamurthy, Professor Dr. Ajit Verma for always encouraging us. I extend my heartiest thanks to our speakers, Dr. Ulrich Mathias and Dr. Rajesh Sharma, for sharing their knowledge and experience with us. My sincere thanks to Dr. Amit Chand Khadakwal for sharing the glimpses of AIMT. My sincere thanks to our teaching and non-teaching staff, students, scholars for their patience and cooperation. My sincere thanks to admin department and IT team for their support and help. And my sincere thanks to ESCO department. Thank you, everyone. Thank you, sir. Thank you, everyone. I would like to request you all to please join us for tea and snacks outside at E2 foyer. Thank you.